Sorry, it cut off again. So we were talking about warm temperature versus cold temperature for phospholipids. Um, and so you know that saturated fatty acids are solid in room temperature, right? And unsaturated fatty acids are fluid in room temperature. Fluidity, again, is governed by the percentage of unsaturated versus saturated fatty acids that you have. And again, cholesterol helps to change fluidity and permeability. The more cholesterol you have, the less permeable you are, and the less fluid you are, because you're like filling up the spaces where those holes and kinks are that allows ions and molecules to go in and out. Um, yeah. Know that in warm temperature, things are more likely to be fluid and vegetable oil like, and at cold temperature, it's more likely to be solid. That's because at warm temperature, phospholipid molecules are more apart from each other. They vibrate a little bit more. When it's cold, they kind of line up more, so it's more solid. I feel like there was a question about bacteria and hot springs, but I don't have a note on it, but I'll just explain it anyways, just in case. There are bacteria that live in or near hot springs where water is very hot. When you look at the phospholipids in their membranes, they tend to be very saturated because you don't want them to be too fluid, which would be very mucky and salts and proteins would pass out too easily. You will find cells have saturated fatty acids. In cold environments, they tend to have polyunsaturated fatty acids. They need all of these kinks and so forth to keep them from lining up and becoming too solid. So in hot springs, you want to have space. You want things to be fluid, but not too fluid, because everything would come out. But in cold environments, even though you're more likely to be more solid, you still need kinks, because if you're too solid, then you don't allow things that you need to come in. Bacteria that live in a puddle in the street and environments. Bacteria sometimes live in a puddle in streets that are hot and cold. When you look at the bacteria that live there, they change the nature of their lipids. So, just like ours, lipids are synthesized and functional, but then they're destroyed and replaced with new ones, so there's turnover. Bacteria, when it's early morning and cold, the lipids have more carbon-carbon double bonds so that they are kinked, and therefore they stay fluid in the cold temperature. And as it starts up, they start to make the lipids more like the saturated fatty acids, which are going to be more butter-like and less fluid-like because they don't want things to spill out everywhere. So that might be confusing. So in warm temperatures, the tendency is for your phospholipids to become very fluid like and push each other apart. But you don't want that to happen. So bacteria, they are able to move their lipids and such so that they become more like saturated acids. so that they are more solid, so things don't spill out to the extreme that you would normally expect at a warm temperature. At a cool temperature, naturally, it's very rigid. That's just what happens with phospholipid molecules. But for the bacteria that they were talking about, because they don't want it to be too solid, at this time they start trying to respond to how solid it might be by making kinks. So hopefully that makes sense. Again, cholesterol, what is its function? It fills the space in lipid bilayers. In absence of cholesterol, membranes would be way too fluid. So it decreases the permeability to, like, the permeability is so extreme, and so it decreases it to, cholesterol decreases it to an appropriate level. Moving on to lecture 6. Okay, so now we are talking about proteins and biological membranes. Danieli and Davison propose a model. It's called the protein-lipid sandwich model for proteins and biological membranes. They propose the proteins would just be on the top and the span of the lipid bilayer instead of having protein within it. So, yeah. So they thought this would be the protein, but it turns out protein are actually within it. That's the Singer-Nicolson model, the fluid mosaic model. So know those two differences. In the Singer-Nicolson model, proteins are integrated into the bilayer. So more on this Singer-Nicolson model. It includes hydrophobic side chains. So the side chains are hydrophobic. And these are from the alpha helical segment, and they extend perpendicular to the helix axis. Again, there should be approximately 20 hydrophobic acids. Of course, there should, and usually you wouldn't find 20, you wouldn't find proline within this chain, but it was talking about you need to have 20 hydrophobic acids to be in this alpha helical conformation to traverse the bilayer. So how many do you need? You need 20. So skipping to the first membrane protein, when we studied it, we saw that there are actually integral membrane proteins that are actually in the phospholipid bilayer, but then there are proteins on the outside, and what do we call this? The peripheral membrane protein. So if the protein is actually in the bilayer, if it's actually within the transmembrane, it is an integral membrane protein. Yep. So we looked at cytochrome proteins. If you wanted to purify, you have to use a detergent. Lipid membrane, components of membrane. Only detergents can solubilize integral membrane proteins. So you have integral membrane proteins, and those are the ones that traverse the bilayer. The only way to get rid of it or to solubilize it is to use detergents. And remember, detergents are amphipathic molecules like fatty acids, and they form micelles. But peripheral membrane proteins, in order to get rid of those, you have to treat them with high salt or extremes of pH. So know the differences. So integral membrane protein, it is in the actual membrane. Peripheral membrane proteins are on the outside. To get rid of the ones on the outside, you need high salt or extreme pH. On the inside, you can use detergents, which are amphipathic and form micelles. There is an important technique that we use. It's called the freeze fracture technique. This freeze fracture technique is when you you froze a cell water mixture, right? And then you use a knife and then you strike it on the side, and you remove the ice water part. In doing that, the fracture splits the lipid bilayer. Then you can study the exterior and the middle of the membrane. And this prepares the surface, the cell surface, for using scanning, for using electron microscopy. So within that, you can observe the middle parts of the membrane, and it contains a lot of pits and mounds in the exterior. Yeah. So it's like freezing the membrane, but then you separate it, and then you can study what's on the inside. And they used the freeze fracture technique to prove that there were proteins on the inside of the layer, like actually integrated in it. Because when they split it apart, they found proteins that were traversing it. Otherwise, how would you know that there would be proteins there? We also need to know that membrane proteins can be glycosylated on their exterior surface, which means they have short chains of sugars attached to them at multiple locations along the exterior part of the polypeptide chain. So this is what glycosylation looks like. So these are proteins, and then the sugar chains attached to the outside. So now we are going to talk about the permeability of membranes and transport of molecules across them. So what is osmosis? Osmosis is the flow of water. What is diffusion? Diffusion is the movement of solutes. What is hypertonic solution? It is a solution that has high solute concentration and low solvent concentration. What is hypotonic solution? It has low solute concentration and high solvent concentration. Water moves from hypotonic to hypertonic because it is trying to equilibralize. Cells in a hypertonic solution will shrink. Water flows out of the cell. Remember, they are flowing to the hypertonic where there is high solute. Cells in hypotonic solution will expand and lyse means like they break, and water flows in. Cells in an isotonic solution you won't see change because they're already in equilibrium. Know how to work these problems, they're very important. He made a note that says you can add solutes together in some situations. Now we're going back to the phospholipid bilayer, but now we're talking about the permeability of it. So it is permeable to 3 different things. 1, hydrophobic molecules, so oxygen, carbon dioxide, nitrogen. The inside tails are hydrophobic, so they can go through that. They are also permeable to small uncharged polar molecules like H2O, water, or glycerol. The third thing that they are permeable, or something that is not permeable actually, are large polar molecules. So if they're small and they're uncharged and they're polar, like water or glycerol, they can pass through. But if they're large and polar, like sugar or ions, then they can't pass through. So the question at hand is, so if sugars and amino acids can't get across phospholipid bilayers, how do they get into cells? And the answer is by membrane proteins that are transport proteins or channel proteins. We have some examples that you guys know. There's a huge difference between active and passive transport. So first, looking at active transport, there are ATP-powered pumps. They pretty much do the work for moving solute from low to high concentration. Usually solutes move from high to low, but active transport goes from low to high. So it could be ATP or powered pumps. Another example is a sodium-potassium pump. It is actively transporting against the gradient. And then these are passive transport. There are ion channels. It doesn't require energy, and it's going with the gradient, not against it. Transporters can actually be passive or active transport. Moving on, in studying the sodium-potassium pump, you need to know the concentrations of various things in and out. One example is that for the sodium-potassium pump, there is a higher concentration of potassium ions inside the cell. So it moves because that's just how it is. So a sodium potassium pump, there is a higher concentration usually of potassium ions inside the cell than outside, and a higher concentration of sodium outside the cells compared to inside the cells. Cells are usually impermeable to sodium, but cells are usually impermeable, so not permeable to sodium, but these ions can leak in, and sometimes the permeability of the cell membrane to sodium briefly increases, so it's more permeable. When that happens, the sodium-potassium pump acts as a pump and gets the extra sodium ions that just came in back out of the cell. Last note for lecture 6 is that in the sodium potassium pump, you are moving 2 potassium ions in, 3 sodium ions out, and it requires ATP. That's all I have for lectures 1 through 6 of my printed notes. I will look for lecture 7, and it actually might be easier if I go over all of the underlined stuff, because you guys haven't gone through lecture 7 yet, and it's better than just the highlights, right? But anyways, talk to you soon. Hope you don't get tired of watching these videos. It's actually kind of tiring to make it, but I hope it helps. If not, we'll try something else for exam 2, but we'll see. Bye.